Welcome to Photosynthesis Decoded, where we unravel how plants capture and move energy around the planet. In this presentation, we'll follow the cellular machinery, the light-driven reactions, and the carbon-fixing cycles that power biomass and ecosystems, while also exploring evolutionary adaptations and engineering strategies that boost photosynthetic performance for climate and food security. This slide shows a chloroplast cross-section and highlights how its internal architecture coordinates energy capture. Light reactions occur in thylakoid stacks, producing ATP and NADPH, which then feed the Calvin cycle in the stroma, where carbon is fixed into sugars. So the diagram ties structure directly to the flow of energy and matter inside the organelle. Here we focus on how pigments harvest photons and funnel that energy to reaction centers. The antenna complexes collect light across a broad spectrum and transfer excitation to photosystems, driving photochemical charge separation and electron flow through carrier proteins and cytochromes, which you can trace across the diagram from photon capture to downstream electron transport. This illustration explains how water splitting replenishes electrons and sets up the proton motive force for ATP generation. Photolysis at photosystem 2 releases electrons, protons, and oxygen. Electrons move along the transport chain while protons accumulate in the thylakoid lumen, and the resulting electrochemical gradient powers ATP synthase to produce the energy currency for carbon assimilation. The Calvin cycle diagram breaks carbon fixation into three coordinated phases. Rubisco attaches CO2 to RuBP, generating 3 PGA, ATP, and NADPH, then reduce those intermediates to G3EP. And finally, a regeneration phase rebuilds RuBP so the cycle continues, with the net output of triose phosphate serving as the building blocks for sugars, starches, and other biosynthetic pathways. This slide tackles photorespiration, the oxygenase side reaction of Rubisco that wastes energy and carbon. The graphic shows environmental factors and evolutionary responses that minimize loss, from high CO2 and optimal temperatures to C4 spatial separation and CAM temporal separation, explaining how plants have evolved strategies to enhance carbon assimilation and resource use efficiency. C4 photosynthesis is portrayed as a spatial CO2 pump that concentrates inorganic carbon around Rubisco in bundle sheath cells. PEP, carboxylase in mesophyll, fixes CO2 into malate, which is transported to the sheath and decarboxylated, raising local CO2 and reducing photorespiration, an adaptation that boosts efficiency under high light and temperature and dominates many tropical grasses. CAM photosynthesis shows the temporal separation of CO2 uptake and fixation to conserve water in arid habitats. Stomata open at night to take in CO2 and store it as malate in vacuoles. Then, during the day, malate is decarboxylated to supply Rubisco, while stomata remain closed, enabling significant water use efficiency compared with conventional C3 metabolism. The Great Oxidation Event panel explains how ancient cyanobacteria transformed Earth's atmosphere by releasing oxygen. That transition shifted the atmosphere from reducing to oxidizing, which enabled aerobic respiration and, ultimately, the emergence of complex multicellular life, illustrating a planetary-scale consequence of persistent photosynthetic activity billions of years ago. This slide outlines global carbon sequestration pathways that leverage photosynthesis for climate stabilization. Reforestation restores ecosystems and stores carbon in biomass. Algae farming offers high-efficiency aquatic capture and scalable biomass. And crop engineering enhances yield and carbon capture, together providing complementary routes to offset anthropogenic emissions and stabilize climate systems. Photosynthesis optimization for food security highlights engineering approaches that reroute biochemistry to unlock biomass gains. The slide presents Rubisco engineering to improve catalysis, carboxysym integration to concentrate CO2 around Rubisco, and photorespiration rerouting to reduce losses, and notes lab advances demonstrating promising biomass increases that could translate into more resilient, productive agriculture. The final slide connects photosynthesis to renewable innovation by showcasing artificial leaves, photovoltaic tandem cells, and algae biofuels. These technologies borrow natural principles like electron transport, light harvesting, and biological carbon fixation to generate fuels and electricity, positioning photosynthesis as both a biological blueprint and an inspiration for sustainable clean energy solutions.